Should be a pretty quick one. Which structure synthesizes proteins for use primarily within the cell? Now, if you've been studying this, you probably remember the thing that synthesizes proteins you should be thinking about is ribosomes. And ribosomes are split into, well, you can separate them into, well, they can separate them different ways. But for now, we're talking about the bound ribosomes and the ones that are free. Where did that line come from? Anyways, bound ribosomes would be found on the rough ER, right? RER. Rough means that there's little dots on them. So here's the rough ER, and I can see the little dots on them. I should use a different color. How about some red here? And you can see these little dots, these little peppered dots here on these logs. These are actually ribosomes, and these ones are bound, therefore they're on the RER. Then you have free ribosomes, which should be dots in the cytoplasm, like I can see some here. And if I look at these, okay, well, what C looks like it's pointing at one, that's a free ribosome. So what's the difference between these? Well, because ribosomes make proteins, if they're bound to the ER, and the ER can pinch off and form these little vesicles, like this little bad boy that you see here. The Golgi apparatus also forms little vesicles. They're already destined to be put into a package and sent off outside of the cell by exocytosis, and then it's going to export the products of those ribosomes, which will be proteins, through translation outside of the cell. So these ones are destined to go out, to party, go out of the cell. And the ones that are made on the free ribosomes are kind of, dispersed throughout the city in the locations where they might be needed. So in other words, over here, any of the ribosomes, the free ribosomes that are creating proteins, they're going to be used primarily within the cell. Let's see what these other things are looking at here. A is the nucleus. B, I guess, is pointing at the RER, the rough endoplasmic reticulum. D, this is a mitochondria over here. This is probably a lysosome. You can never be too sure about the lysosomes because they just look like larger vesicles. So you really need to know what kind of enzymes are inside, and the sizes can vary as well too. So I never really see this as a correct answer to be chosen. But anyways, hopefully you understood that, which structure synthesizes proteins for use primarily within the cell. Again, use this opportunity to branch out about proteins. What else can you say about proteins? You've got different levels of structure for higher level students: primary, secondary, tertiary, quaternary. What's going on for there? You've got fibrous versus globular proteins. You've got proteins being made by ribosomes. Oh, earlier, for those of you who still are listening, we said ribosomes can be split up in different categories. Remember, in a eukaryotic cell like this, we say the size is 80 Svedberg units. And for prokaryotic cells, we say it's like 70 or 70 Svedberg units. For higher level students, you should be able to draw what a ribosome looks like as well too. If you remember that, large ribosomal subunit, small ribosomal subunit. The mRNA just kind of sits along here, and you've got 3 binding spots called A, P, and E for the tRNAs to come in. But everything's linked together, so don't be afraid to try and go over the top while you're using these questions to study and prepare yourselves. Okay, I'm not going to say alrighty.